Okay, so this video is going to be about the last project of the semester, and this project will take us from this week all the way through the end of the semester. Um, and it's built around trying to identify an unknown algae. Now, this project is not hypothesis driven. This would be a part of a larger experiment that somebody would conduct, and I'll talk more about what that might be when we get into lab. But for now, the whole project is not hypothesis driven. That means you don't need hypotheses for your lab notebook. So up front, you're going to get a sample of unknown algae, and it'll be your job to determine exactly what genus and species of algae you have. Now, this algae could be regular old green algae. It could be blue-green algae. It might be diatomaceous algae. That pretty much encompasses all organisms that we consider to be algae in biology. And trust me when I tell you that there's hundreds of thousands of species of algae across these three categories. So how are we going to decide what the genus and species of our unknown is? How can we possibly identify them out of those hundreds of thousands of possibilities? Well, one way would be to look at it under a microscope. And that's how you did it when you were in Biology 181 lab. You looked at algae under the microscope and compared it to pictures and tried to decide what it was. You may have noticed in that lab that some of the algae looks alike. And when you think of the hundreds of thousands of types of algae out there, a lot of them look very, very similar. Small, green, round, hard to tell apart. That's not the only way they look. There are other looks, but a lot of them look very, very similar. From that alone, we probably wouldn't be able to determine genus species level. So one way to look at it is to think to yourself, what makes us different from chimpanzees? Besides the fact that we drive cars. It's the differences in our DNA. In fact, our DNA differs from chimpanzees by about 1.2%. But those differences can tell us a lot about those two organisms and how long ago they separated evolutionarily. So increasingly, labs around the country are turning to DNA to determine the tree of life or the relatedness of organisms. The techniques that we're going to employ are common in molecular biology for doing all sorts of different things, but they're also being used in ecology laboratories and evolution laboratories for studying how organisms are related. So these techniques are in broad usage and are very, very powerful techniques. Since we've been doing this, there are databases that have been built of known DNA sequences. In other words, DNA sequences from known organisms. And so, for example, if you looked at the DNA sequence of human hemoglobin and the DNA sequence for chimpanzee hemoglobin, you could look at the differences and you could see how different those two genes are. Some databases contain whole genomes. The Human Genome Project, a genome is all the DNA inside the nucleus of a cell. So in other words, it would be all 23 of our chromosomes. Remember that we have 2 copies of 23 in each of our cells. So all 23 of our chromosomes have been sequenced. That means we know the order of the bases A, C, T, and G, the order in which they occur in each of the 23 human chromosomes. And this has been done for all sorts of different organisms. In addition to whole genomes, a lot of databases contain specific gene sequences. Again, if you wanted to compare the gene for hemoglobin in humans to the gene for hemoglobin in sheep, you could do that. Those sequences are well known and they're in the database, or a database. This, of course, would include a lot of different algae species. So someone out there has sequenced, or a lot of people out there have sequenced genes from algae, and we can compare our gene from an unknown algae to the known DNA sequences and figure out exactly what we've got. So all we really have to do is get a hold of a specific part of our unknown algae's DNA. Not the whole genome, just a specific part of the genome. Then we determine its sequence. What order do those A's, C's, T's, and G's fall in for our gene of interest? Then compare our unknown sequence to the sequences from known algae species that are in the databases. And again, there's a lot of different databases out there. We're going to use a fairly common one in our experiment. Once we do that, we should be able to determine what the genus and possibly even the species of our algae is. So that sounded pretty easy, but it's never quite that easy, and it is going to take us till the end of the semester. So let's look at the broad outline of steps that we're going to use. First, we're going to isolate genomic DNA from our unknown algae. That means all the DNA from all the nuclei in all of the algae cells in your sample. Then we're going to make a lot of copies of a specific gene of interest. And by a lot, I mean billions of copies of our gene of interest. So just that one part of the genome, we're going to copy billions of times. This is called amplifying the gene. And the process that we do it by is known as polymerase chain reaction, and I have a whole video ready to go for that, as well as I'm going to give you a link to a video as well. Once we have billions of copies of our gene of interest, we're going to send that out for sequencing. We don't have quite the right equipment on campus to do the sequencing ourselves, and it's much easier for labs that just, this is all they do is sequence DNA. So they can do it for us in a week. We'll get the answer back. We'll get the sequence back from them. We're going to use a process that's really a modified Sanger sequencing, and there will be a video in the future about that. Then finally, once we've got our sequence back from that site, we will use a form of bioinformatics to compare our sequence to known sequences in the database. And that should give us our answer as to what kind of algae it is.